Hello, APBio. Welcome to our video lecture for Chapter 32, Homeostasis and Endocrine Signaling. So in the last video, I began by saying that the two big topics that we're addressing in this series of chapters is, one, how cells communicate with one another, and two, how organisms respond to internal and external changes. And this chapter being on hormones and the endocrine system really hits both topics. Cells communicate through hormones, and cells respond to their surroundings through hormones. So let's dig right in. So regulating versus conforming. So there's many different examples of what I mean by these two terms. So faced with environmental fluctuations, organisms can either, they have to choose, are they going to regulate their own internal conditions, or are they going to conform to what the external environment is like? A regulator, let's use average body temperature as an example. A regulator maintains a 98.6, thereabouts, the actual number is debatable, body temperature regardless of what's going on outside. It could be 7 degrees outside, it could be 100 degrees outside, your inside is going to be 98.6, give or take. That's what a regulator does. The benefits of being a regulator is that you can maintain it yourself. You don't rely upon the outside. The cost is it's very energetic. It takes lots of calories to maintain a body temperature of 98.6 degrees Fahrenheit. So you have to eat a lot more than things that are ectotherms, that they can't maintain their own body temperature. A conformer in our case would be something like an alligator, where whatever the temperature is outside will be its temperature inside. The advantage would be, the disadvantage would be you can't regulate it yourself. You have to rely upon the environment. But the advantage would be you need way less energy to maintain that. So if an alligator is too hot, it can just go into a body of water and cool off. If it's too cold, it can go bask on a rock. So a regulator is better in that you can control it yourself. A conformer is better in that you don't need as high an energy intake to maintain the conditions. A graph like this is showing the ambient environmental temperature on the x-axis and the body temperature on the y-axis of a river otter and a largemouth bass. A river otter is an endotherm. It maintains its own body temperature. It is a mammal. And a fish obviously is an ectotherm. Notice I'm not using the terms cold and warm-blooded. Why that is, we'll see in a minute. So if the AP exam gave this to you and said which one's the regulator, so you had to look at what Y is doing over X. So X is the outside. Is the outside changing as you go left to right? Absolutely. Is the Y changing with the river otter? No. So it's regulating the body temperature. Is the largemouth bass changing left to right? It is changing, so it is a conformer. All right. If what's happening outside matches what's happening inside, or vice versa, it's a conformer. If they don't match, if what's inside is being constant, it's a regulator. So homeostasis, you've heard this term before. It's just a fancy way of saying steady state. So it's your body's ability to maintain steady conditions inside, regardless of what's going on outside. So in humans, body temperature, blood pH, blood glucose are all maintained at a pretty constant level. If you look at this diagram, the analogy that most textbooks use is like a thermostat. So in the middle is my set point. So say it's set at 20 degrees Celsius. If it gets too hot, there's a signal to bring it back down. If it gets too cold, there's a signal to bring it back up. So your body temperature, 98.6 or 97.5, depending upon the source you're looking at. Blood pH around 7.6 or so. Blood glucose, we'll do that example on a later slide. There's feedback pathways that if it gets too high, you bring it down. If it gets too low, you bring it up. Your body's always looking for homeostasis. It's always trying to define normal through these feedback paths. So this slide is kind of what I just said. So there's a set point, all right? If a stimulus brings it up too high or too low, some kind of sensor detects that, and the response is to bring it back down to homeostasis. This is called negative feedback. This is super important. Negative feedback means if it gets too high, it comes down, gets too low, it comes up. So negative feedback reduces fluctuations. It reduces the stimulus, all right? Your body tries to counter whatever is happening to take it away from homeostasis. Most of these feedback pathways in the body are negative feedback. There's a couple examples of positive feedback, which we'll get to on a later slide in this chapter. But body temperature, blood glucose level, blood pH, it's all negative feedback. Thermoregulation. So I said a minute ago, we don't use the term warm-blooded and cold-blooded. Something like a fish that you might say is cold-blooded. Well, if it's warm outside, its blood is warm. So that term is kind of a misnomer. Endothermic and ectothermic is the term that we use. So thermoregulation just means how do you regulate your internal body temperature. And endotherm keeps it stable. So that's what we would call warm-blooded back in the day. If it gets too hot, you sweat. It gets too cold, you shiver. An ectotherm cannot regulate its body temperature internally, so it has to do so by behavioral means. That lizard, if it gets too hot, go in the water. It gets too cold, go sit on that rock. And the benefit of being an ectotherm, again, is you consume way less food because you're not regulating your body temperature yourself. Another way to regulate thermoregulation, so vasodilation and vasoconstriction, this refers to the diameter of your blood vessels. Your body can make the blood vessels wider or narrower. And particularly talking about your superficial blood vessels, the ones near the surface of your skin. And what you need to know, vasodilation means make them bigger. Vasoconstriction means make them smaller. And if you make them bigger, if they dilate, the blood vessels lose heat. So say, you're getting hot, your face gets flushed, right? Well, your face turns red because your blood vessels are dilating. You're getting more blood flow to your cheeks, which helps irradiate out that extra heat. If you get too cold, your blood vessels constrict, which helps to reduce heat loss so that you don't get hypothermia. This idea of countercurrent heat exchange, so let me define it, then I'll show you, I'll make this picture bigger. So having fluids running in opposite directions is a way of minimizing heat loss, and it's a way of maximizing the transfer of heat from one direction to another. Let me show you this picture bigger. This is showing arteries and veins in a goose's leg, its feet, which get very cold. So you notice here I have an artery, the blood flow is going down. Here I have a vein, the blood flow is going up. Look at the temperature variance, 35 to 33, 30 to 27, 20 to 18, 10 to 9. Heat is always being transferred to the right, all right? And the variance, that's what, 3 degrees difference, that's 3, that's 2, that's 1. The temperature differences are very, very small, but the way that it works, because the blood flow is countercurrent, it's always going from left to right. If it was, if it went left to right up here, right to left down here, eventually it would all mix together and it wouldn't be an efficient transfer of energy. Veins are usually on the outside, like further superficial than arteries. Arteries are usually deeper in an organism, so your veins are going to be more exposed to the cold. So the idea is you're transferring the heat from arteries to veins more efficiently because the blood flow is going different directions. There's lots of examples of this in the body where it has fluids running countercurrent. It's always to maximize the exchange of heat or sodium or glucose or whatever from one vessel to another. Okay, so now let's get into the endocrine system. So your body has two main communication signals that it uses to communicate. There's the endocrine system and there's the nervous system. The endocrine system is the system that uses signaling molecules called hormones. We discussed this in chapter 31 with plants. So compared to your nervous system, this is it's a slower system. It's by no means slow, but nerves work faster, and it's a longer lasting communication system. So hormones, you have hormones that might be released by your brain that affect your kidneys, or hormones that are released by your pancreas that affect your intestines. When an organ releases hormones, they go into your blood, they go throughout your entire body, but they only work on certain cells, certain target cells that have the receptors in their cell membranes for that hormone, right? If the hormone, testosterone affects sweat glands, that's because, and I'm making up the example, that's because the sweat glands have the receptors for testosterone. If testosterone does not affect my rib cage, it's because the cells of my rib cage don't have the receptors for testosterone. I made up those examples. They might not be right, but you get the idea. All right. It's a longer lasting system because the hormones, they don't last forever, but until the hormone gets broken down, the effect is still happening. The second system, which this is a later chapter, your nervous system, uses electrochemical signals. It's almost like sending electricity down a cord. Nerve impulses go faster than endocrine system impulses, and you can turn them off quicker. We'll discuss the nervous system obviously in a later chapter. So this just reviews the two. The endocrine system release hormones that get into your blood, they go to target cells. The nervous system uses nerves, these long wire-like cells called axons, or the parts of a cell are called axons, for using electrochemical signals. Okay, so let's just go through an example. You don't need to memorize this, but the AP exam could give you examples to analyze, so let's just do one together. So this is a simple endocrine pathway. So your stomach has a very, very low pH. You probably already know that. Digestive juices in the stomach are very, very acidic. When food leaves your stomach, it goes into the small intestine next. The first section of the small intestine is called the duodenum. Some folks pronounce that duodenum. I was taught duodenum in school, so I stick with duodenum. So the duodenum does not like it that acidic. The acidity coming from the stomach into the small intestine needs to be at least partially neutralized. And what happens is when the contents of the stomach enter the small intestine, there are endocrine cells actually in the small intestine that secrete a hormone called secretin. And secretin gets into your blood and eventually goes to your pancreas and causes the pancreas to release bicarbonate ions into the duodenum. So this is a negative feedback pathway. When the pH gets too low in the small intestine, it releases a hormone whose end result is to bring the pH back up, all right? Again, don't memorize this. This is just one where if the AP exam gave you this diagram or this text as a paragraph and asked you questions about it, you would understand what's going on. This is just the same diagram, bigger. And it gets negative feedback because the pH got too low and then you brought it back up. So this chapter goes through all the different endocrine organs, which we don't need to do this for the AP exam. I put in this picture just to show the endocrine system is basically scattered throughout your entire body. There's little patches of endocrine tissue in lots of different places. None of this do you need to memorize, but it's interesting just to look at to see where the things are that we're talking about. So there are a handful of hormones that I want you to know what they do. Some of these hormones are produced in what's called the posterior pituitary gland, which is in your brain. And some are produced in what's called the anterior pituitary, which is also in your brain. So all 8 of these hormones, are made in your brain, and they go all over your body through your blood to do different things. This 8 that I want you to know. So oxytocin is a hormone that stimulates uterine muscle contractions during childbirth. ADH is called antidiuretic hormone. A diuretic makes you urinate. So ADH, antidiuretic hormone, makes you urinate less. It tells your kidneys to conserve water. Your kidneys filter your blood, and what comes out of your kidneys becomes urine. So if your kidneys are conserving water, you're urinating less. Growth hormone, the name gives it away, causes you to grow. Prolactin is a hormone that regulates milk production in lactating women. FSH stands for follicle stimulating hormone. It's made by the brain, targets the ovaries. It's a hormone that we're going to see in chapter 35, so we'll come back to it. LH stands for luteinizing hormone. It's also produced by the brain, targets the ovaries. It regulates ovulation in a woman. We'll come back to that hormone in chapter 35, I believe. TSH is called thyroid stimulating hormone. Your thyroid gland is in your neck, and it regulates largely your metabolism. And ACTH is a hormone that is made in your adrenal glands on top of your kidneys. It has a wide variety of functions. In this case, I just want you to know that it stimulates your adrenal glands on top of your kidneys. So all the examples we've done so far are examples of negative feedback. So let's do 1 or 2 of positive feedback. Positive feedback would be something where a stimulus happens and the body's response is to reinforce the stimulus to happen even more. There aren't a whole lot of examples of this in the body. The 2 that the book usually mentions are childbirth. When the uterus is contracting during childbirth, the body's response is to cause the contractions to increase. Or when a baby is breastfeeding, the suckling reflex stimulates the breast to produce even more milk, and the woman will keep producing milk until the suckling reflex stops. Okay, so this is the 3rd time we've done this topic in AP Bio, so hopefully you can tell this is a super important topic. Signal transduction pathways. We did this in this chapter on the cell membrane, then we did it again when we were talking about cell division, I believe. So there's two main schemes. This first slide goes through the first scheme, which the second scheme is the one that I really want to focus on. So look what's happening in this picture. I have a hormone, in this case testosterone. It actually passes through the cell membrane, passes through the nuclear membrane, binds to a receptor on DNA, and either turns the gene on or turns the gene off. The key to this scheme is the hormone actually enters the cell. Testosterone can do that. Most hormones, though, the second scheme is the one on this slide, and you've seen this picture before. Most hormones don't actually enter the cell. They bind to a receptor on the plasma membrane. So look at this picture, and we've seen this exact same picture before. I have the hormone. Here it's just called a signaling molecule, this guitar pick-shaped molecule. It binds to a likely protein receptor in the cell membrane. That's called reception, all right? The second stage is called transduction. This is when the signal gets carried and amplified through the cytoplasm. Do you remember back from the chapter on the cell membrane, we discussed protein kinases and protein phosphatases. Protein kinases added phosphate groups to these intermediates, and it largely makes them change their shape. That's how you get the signal from the membrane to the nucleus. The third stage is called the response. This just says activation. It's likely turning a gene on or turning a gene off. The three parts of signal transduction, it's super important that you understand these. This is a different picture of this scheme. This shows a growth factor. So this is reception. Notice in transduction, it shows a phosphorylation cascade. The word cascade is a great word because I could have the hormone bind once or bind to one receptor, and this could be going on hundreds of times. So not only does it get the signal to the nucleus, but it can also amplify the signal to where only a little bit of hormone can have a huge impact. Let me ask you this question real quick. What would happen to this whole thing if I mutated the receptor and changed its shape? If the receptor had the wrong shape, such that the growth factor couldn't bind, the signal doesn't get sent to the nucleus at all. The signal, in effect, it gets turned off. The third stage again here is the response. This just shows a transcription factor that was inactive, and the signal turned the transcription factor on, which made the gene be transcribed, which obviously the gene's transcribed, it's going to be turned on. In the chapter on mitosis, when we did those checkpoints, so remember we had tumor suppressor genes that slow growth down. Well, there are molecules, so each of these sections in this cascade is catalyzed by an enzyme, and there are molecules that block those enzymes. So if a tumor suppressor gene would be blocking this, which would stop the signal. If your signal is to grow and I stop the signal, the cells don't go through mitosis, so the cell doesn't divide. This is a way that cells can prevent becoming cancerous by having tumor suppressor genes. Proto-oncogenes were ones that were like the gas pedal. Those were ones that caused the cell to divide. A proto-oncogene could be a gene for one of these proteins in the phosphorylation cascade, where if I overexpress that and I have too much of this going on, one growth factor signal could cause way too much growth because I'm overdoing this. So in cancer, we've discussed how it's a combo of the gas pedal genes, the oncogene, or the proto-oncogenes become oncogenes, and the tumor suppressor genes, which are like brake pedals. If I mess with the brakes, you can't slow the car down. If I mess with the gas pedal, you're going to go too fast. So it's two different ways of regulating growth, either turning it on or slowing it down. But all of it involves these signal transduction pathways, which is why, I'm going to go back, we've done this slide like 3 times already. The AP exam loves signal transduction because they can use it for examples. I mean, from the cell membrane to cancer to mitosis, you name it, it just loves signal transduction pathways. This slide just gives an example. So oftentimes hormones have multiple effects. Epinephrine, which is similar to adrenaline, is produced by the kidneys. So you would produce adrenaline, like if you're stressed, if you're running, if the robber's chasing you with a knife, you get a burst of adrenaline. Adrenaline or epinephrine, look at what it says. It increases blood flow to your muscles, which will help you run from the murderer. It decreases blood flow to your digestive system. You don't want to, be digesting a food and having, an upset stomach because you're running from the murderer. So it kind of slows down your digestive process. It also increases your blood sugar levels so that your muscles have a good source of energy. And you can see epinephrine, that's the red circle. The little U-shaped things are different receptors. Liver cells have glycogen stores, which if you break down glycogen, you produce glucose. Liver cells have the receptor, and then blood vessels have the receptor. In this case, to dilate them so you get more blood flow. And actually, according to this, epinephrine can also, so it's dilating blood vessels in your muscles, and it's constricting blood vessels in your digestive system. So you don't have to go to the bathroom when you're running from the murderer, okay? So what dictates what cells respond is, do they have the receptor, in this case for epinephrine? So we're gonna wrap it up. So I wanna go through just one quick example of glucose homeostasis. So, you know, we discussed one pathway with body temperature. You get too hot, you sweat, get too cold, you shiver. With blood glucose, so about 70 to 110 milligrams of glucose per 100 mils of blood. The number's around 100 or so. That's a typical blood sugar level. If your blood sugar gets too high, say you just ate a meal, and so your blood sugar is high because you just ate. Insulin lowers your blood sugar. So where does the sugar go? Insulin causes cells to take in sugar. So when your blood sugar gets low due to insulin, it's because the sugar is going from your blood into your cells, maybe to store it. If your blood sugar gets too low, you have a hormone called glucagon. So if you have excess sugar in your blood, your body largely stores that, or one place it stores it is in your liver. Your liver has these glycogen stores, which I mentioned like 3 minutes ago. Glucagon causes your blood sugar to go up because it tells your liver to break down glycogen into glucose and release it into your blood. Both these hormones, insulin and glucagon, are produced in the pancreas, and they're called antagonistic hormones because they do opposite things. There are lots of examples of hormones that come in pairs that do opposite things. I want you to know one. So here's a great picture. I think I asked you to draw this in the lecture outline. So you can draw a simpler version of this, but the middle is about 100, between 70 and 110 milligrams of glucose per 100 mils of blood. Blood sugar gets too high, you produce insulin, brings it down. Gets too low, you produce glycogen, which brings it back up. All right, this is negative feedback because if you get too far away from the middle, you're going to bring it back down. This slide just mentions the two types of diabetes. You don't need to know this for the AP exam. Type 1 is an autoimmune disorder where your immune system targets your pancreas, which is not something that you want to have happen. Type 2 diabetes is when either the target cells, well, for whatever reason, the target cells aren't responding to insulin. There's lots of lifestyle factors. Things like being overweight can lead to obesity. There's factors that are genetic. If you have type 1 diabetes because you're overweight, if you lose weight, the diabetes can be managed. So it's largely a lifestyle disease, type 2 diabetes. I want to end with just a note on osmoregulation. So we talked about this when we did the chapter on osmosis way back when. Osmoregulation is basically an animal. Think about things that live in the water like fish, how they balance the solute concentration inside their cells. Osmolarity is a fancy word for the solute concentration of a solution. So if it's 10 molar sodium chloride, the osmolarity is 10 molar. Don't let the word confuse you. If two solutions have different osmolarities, water follows solute. This third point uses the term hypoosmotic and hyperosmotic. We use the terms hypertonic and hypotonic back in the day. Water's going to go where solute goes. Hypertonic or osmotic means there's more solute. So water goes from hypoosmotic to hyperosmotic, or hypotonic to hypertonic. It's really the same thing. If I put a saltwater fish, like a clownfish, which is used to salt water, put that in fresh water, it's going to gain water. It's going to be a problem. If I put a freshwater fish into salt water, it's going to lose water. Its cells will shrivel up and die. So things like fish have adaptations to prevent this. Marine fish are always losing water to the ocean, so they drink a lot. They drink water, and they have special features in their gills where they can excrete the excess salt. Freshwater fish, they're always taking in too much water because they live in freshwater. They don't hardly drink at all. They have very dilute urine. They pee a lot, and they can replenish the salts they need through eating. So things that a marine fish can do to prevent losing too much water. They drink a lot, they excrete the excess salt, and their urine is very concentrated. They don't pee a whole lot. Freshwater fish, what do? They don't drink. They have a very dilute urine. They pee a lot, and they can replenish salts through things that they eat. Okay, so that was a lot for one chapter. We're going to call it a day there. Hope that was helpful, and I will see you guys next time.